is a very world-renowned, I will say, international and domestic minimally invasive breast surgeon. And she is a new inventor with a um, pre-venture that she is working on. And Rasha, over to you.
Well, thank you, Donna. Let me share my screen. It's a little quirky yesterday, so let's see how this goes. Let's see. So, all right. Can you see my screen?
Perfect. Looks great.
Okay, perfect. All right, good. So, first of all, thank you for the invitation to give an overview today of my invention, a flexible cavity filling device with fiducial markers for patients undergoing lumpectomy. So, just a brief introduction to the team. I'm a professor of surgery. I've had more than 30 years of clinical experience. And it was really through that experience treating breast cancer patients that I came upon the need for this device, as well as my experience working with device companies gave me the idea of some options of how to create something that would work. It's through that device company experience that I met the second team member, Anthony Carnamola. He and I worked together years and years ago, it was decades ago, on a consulting arrangement that I had with a surgical device company. And then Donna, part of our CTL team. And then we also have been working with a medical device development company, VitaTech, and they really have been instrumental in helping us identify the materials and helping us create our prototype. So just as an introduction, I think it's important for us to recognize that breast cancer is a really common disease. I mean, just in this country alone, we're going to have over 300,000 new cases of breast cancer. And I would venture to say there isn't a person in this meeting that doesn't know someone who's had breast cancer. It's just such a common disease. Now, most of the patients will end up having a lumpectomy, a partial removal of breast tissue. And there's also a role for lumpectomy in benign tumors as well. So you can see here the numbers, they're really big. Now, some people say, well, what's a lumpectomy? So a lumpectomy is a surgical procedure where the surgeon would remove an amount of tissue that would be including the tumor, either benign or malignant tumor. And the concern is that sometimes when we remove tissue, depending upon the volume that we need to remove relative to the size of the breast and other various clinical factors, it can leave a pretty significant distortion. And these patients, of course, the most important thing is to cure them from their breast cancer. But we're pretty good at that, actually. So these are patients that are going to live for decades after their breast surgery. And we really don't want them to have this kind of a physical deformity after they've had their lumpectomy. You can see here in this picture, this is a patient who had a lumpectomy and has a significant indentation, of a sinkhole in her breast, so to speak. Now, the other thing that we really need from a clinical standpoint is we need to be able to mark the area where the surgeon performs a lumpectomy, either as a target for radiation that often is a partner with a lumpectomy for cancer, or just to designate the area where the patient had the surgery for future imaging. And you have to mark it, but that has to also be balanced with the fact that we can't obscure the area, because we are going to need to follow these patients very carefully from an imaging standpoint for recurrences. So the existing options that are available clinically really are suboptimal. One of them is this rigid basket that is hard, and patients don't like the fact that it's hard. When it degrades, it sometimes just collapses, and that defeats the whole purpose of putting it in from filling the space standpoint. But also sometimes I've seen that when it degrades, it doesn't just melt away, it breaks down to these sharp shards of tissue that can be painful for the patient. And this device has actually been pulled off the market. The other available option are these radiopaque or metallic sutures. Well, we can see them on imaging, but they don't fill the space. So that really doesn't satisfy the criteria of what we need. Now, silicone's been around for a long time, but the problem with silicone is it's not radiolucent, it's not transparent on imaging. So to put in a silicone device would actually obscure the area on imaging and would not allow us to follow the patient for recurrences in the future. So what's the unmet need? We need something that is permanent, that it fills the volume. It needs to be customizable because these lumpectomies are various sizes. It needs to mark the area on imaging, but at the same time not obscure it on imaging. It needs to feel like breast tissue. It needs to be soft. I need to be able to secure it in place so it doesn't migrate in any way. If I ever need to go back, I want to be able to retrieve it very easily, and it needs to be able to withstand radiation therapy for these breast cancer patients. So here is my device. Think of it as a strand of pearls. It's a strand of pearls with these radiolucent, transparent polymer beads. It feels just like breast tissue. I can adjust it to fill whatever size cavity I need to. All I have to do in the operating room is just trim the strand. I can use 5 beads, I can use 20 beads, I can use 40 beads, depending upon the size that I need to fill the cavity. It's permanent, so it's not going away. It stays in place. I can just suture it into the cavity and have it stay there. And if I ever need to go back, I can just pull it right out. It has, if you look at this close-up view, every so many beads, this tiny little metal marker. And that little metal marker is what's going to be seen on imaging and allow us to know where the cavity is and also allow for a target with radiation. We do have several patents. We filed another one yesterday, as a matter of fact. Here you can see in the bottom image, I'm holding it in my hand. It's this soft little bead on a strand. And again, it feels just like breast tissue. The lower right image is really important. What you're seeing here is this upper tissue is a breast. That's a breast on X-ray mammogram. And if you look at the beads on the left, those are silicone. You can't see through those. Whereas if you look at the beads on the right, you can see through them. You wouldn't probably notice, but half of that strand is underneath the breast, where that little white bar is. That's a marker inside of this strand. So you cannot even see this on mammogram. It looks just like breast tissue. So again, the market is huge for just the United States. The other thing we could use this for would be not just in the time someone has a lumpectomy, but there are many, many patients who now have this deformity from a previous lumpectomy. This could be used for revisions as well. And it's not just something that we would use in breast. This could be used in any kind of a soft tissue defect. This could be used for sarcoma surgery, head and neck surgery, the list goes on and on. So again, the market is really sizable. So here's where we are. I was very fortunate to have a grateful patient fund that allowed us to get through phase 0, phase 1. So we're really now into phase 2, 3, where we have a prototype, but we're working on getting a much more high fidelity prototype, doing some more of our preclinical, maybe an animal model, and then as you can see here, going on to 510K and then eventually to commercialization. So here's the ask, $1 million. And the use of the funds would be the product development, taking it further. The preclinical, a lot of that is actually very straightforward because what I'm doing now is I'm taking these prototypes. As you saw the image before, I can just get x-rays in the operating room of a breast, a real human breast, with these next to it. I can simulate a lumpectomy in a human breast that's been removed, and then I can get x-rays on this. So that part is essentially free. So that's a wonderful resource to not be spending money. The animal model, I don't know if we're going to have to do an animal model. We might, but hopefully not, because every single piece of this device, all the components are something that has a long-standing history of safety in human beings. So hopefully we won't need to do an animal model. If we did, I could take a little bead, put it in a mouse, leave it there for 3 to 6 months, take it out and just show that it didn't really do anything. And we're moving along very quickly, actually amazingly quickly. And we really think that it is realistic that we could be at the point of a first in human clinical trial in the next 18 to 24 months. So I am going to stop, thank you, and I'm going to stop sharing my screen. And I'm going to show you the actual device. Let's see, to go back. Okay, great. So this is a prototype, an early prototype. That's silicone. Now we're not using silicone because you can't see through silicone on X-ray. But just to give you an idea, I mean, you see how flexible it is. I mean, if this is a lumpectomy, I can just put it into the lumpectomy cavity, and then I can just pull it back out if I ever need to. You probably can't see, but every few beads, there's a tiny little metal marker here that's actually what we're going to be using. Now, this is the real material that we're going to be using. Very rough prototype, but here it is. This is more of the size of the beads, and look how squishy it is. It's just like breast tissue. So with that, I'll be happy to answer any questions.
Fantastic. Thank you, Rache. Do we have any questions for Rache?
Yes, I think there's 3 new Q&A questions.
Brilliant. So, what do you mean by philanthropic funding?
Right. So far, we've been very fortunate that we've been able to have undiluted funding of our IP. So it would be fantastic if we could continue that way. We are realistic in the fact that we know at some point we'll be licensing this, and we may be partnering before we license it. So all of those options are on the table for discussion.
So philanthropic funding, just to add a flavor, is additional grateful patient funds. So those are tax-deductible funds that you can provide to Rache through Weill Cornell Hospital. And that would be a great way. Non-dilutive funding is an important way to get these early stage technologies up and running. So who determines the need for animal testing?
Well, that will really be regulatory. You know, if we can't take it to a first in human without animal testing, then we obviously have to do it. And we'll just have to see. We can certainly speak to them and just say, what do you think? Are we going to need to do animal testing? If A is safe in human and B is safe in human, putting A and B together should be safe in human. That may be all we need to do, or they may insist that we do, again, a mouse model, which would not be a huge deal to do.
Right.
We don't need to do this on large animals like, you know, pigs or whatever. So a mouse would be fine.
Very good. And then do you have a commercialization strategy in place?
Not yet, but we are discussing it. Yes.
We are indeed discussing it. So this is a ongoing opportunity. Let's see. Any other questions? Great presentation. If it's an implantable, wouldn't it be a PMA pathway? And then how do you sterilize this? Is this gas or gamma?
Yet to be determined, but obviously would have to be sterilized to put in a human being, you know, of course. So that's yet to be determined.
Yeah. And then just the other question, the implantable, wouldn't it be a PMA pathway? No, actually not. So for example, total knee replacements are all class 2 devices. You don't need, as long as you have a predicate device. And so with regard to this one, we are certainly looking for what the predicate device would be for the FDA. Okay. Awesome presentation. Any other questions? Okay. Rache, let's give Rache a big hand, and we will go on to our